Gentlemen, my name is Enrico Scala from the Istituto Dermopatico dell'Immacolata, and I'd like to present our last study entitled HV1 and number 4 co-sensitization identifies Italian patients at risk for Mugwort celery spike syndrome. Pollen defenses, particularly HV1 for Mugwort and number 4 for ragweed, are key allergens involved in pollen food syndromes. Despite their recognized molecular relevance, their clinical impact in southern Europe and especially in Italy has remained poorly defined. Comparative amino acid alignment of RTB1, MB4, and APG7 revealed a broad family of defensin-like protein across multiple plant species. Homologous sharing 24 to 66% sequence identity with RTB1 were identified in foods and pollen such as peanut, carrot, celery, sunflower, lupin, soybean, pepper, and poppy. Some homologous corresponded to established allergen, for example peanut and horse chestnut, while others remain uncharacterized, suggesting a wider and still underestimated defensin-driven cross-reactivity network relevant for clinical allergy. Among more than 7,000 patients tested in the last 5 years, 3.8% were sensitized to at least one pollen defensin. Most cases showed monosensitization, predominantly to RTB1, typically associated with respiratory allergen. In contrast, dual sensitization to RTB1 and ABA4 identify a distinct, different subgroup. In a defensin-sensitized subset, additionally tested for APG7, more than 80% showed IgE reactivity to celery defensins. Co-sensitization was strongest between RTB1 and APG7 with a robust linear correlation of IgE levels. By contrast, recognition of other allergens from celery, for example PR10 or non-specific lipid transfer proteins, was less frequent, highlighting defensins as the dominant molecular link between pollen sensitization and celery-related food allergy in this population. Functional inhibition experiment demonstrated a clear immunological hierarchy. RTB1 acts as the primary sensitizer, driving IgE cross-reactivity towards both MB4 and APG7. Clinically, dual RTB1 and MB4 sensitization was strongly associated with systemic food reactions, conferring a nearly seven-fold increased risk compared with monosensitized individuals. In conclusion, defensin sensitization in Italy reveals distinct molecular and clinical signatures. While isolated RTB1 marks a respiratory allergy, co-sensitization to ABA4, often accompanied by APG7 recognition, identifies patients at higher risk for food-induced systemic reactions, supporting a precision medicine approach in pollen food syndrome. And thank you very much for your so kind attention.